This is chemical characterization, how to prepare for the future in this time of transition. I think that this is a really exciting time for biocompatibility and chemistry both, as both the things, both subject areas have been evolving pretty rapidly over the last couple of years. So it's really timely to be getting into this now. This landscape, honestly, it's been changing just within the last couple of months. So we see, new trends coming from the FDA all the time, the types of things that they are interested in, what they're asking for, and so on. The strategy when it comes to biocompatibility is really, shifting. We made a big change 2 years ago, 3 years ago, when we really started to do a risk-based approach to biocompatibility. Before that, we would just go down and checkbox all of these different risks. We'd say, okay, this is the device, this is the contact duration, so these are the risks, and you just test all of them. But the FDA, part of their mission is to try to provide the least burdensome solution to getting devices to market, and I think that they've taken that a little bit to heart. And as part of that, we've started to see now them being pretty reasonable when it comes to accepting good scientific rationale and things like that. So it's a great time to be doing this. Both of these topics, so I'm talking about chemical characterization today, I'm talking about biocompatibility tomorrow. Both of these topics are really intimate with each other. 45 minutes is not nearly enough time to really cover any of these topics with any justice. So feel free to ask any specific questions that you might have throughout the talk. And we do the same thing that we're doing here, but over 3 days throughout the year. So this is something that Nielsen Labs does on a regular basis. The next time that we're going to run one of these seminars is in September in Chicago. So if you just can't get enough of this type of chatter, then you can think about going to one of those. Okay, so before I get going, let me just first give a brief personal introduction. So I basically grew up under a rock reading books when everybody was out, making friends and getting sunlight on their skin. I was alone in the basement with my calculator, I went into to academics, got a PhD in physical chemistry, went on, I taught chemistry and quantum mechanics for a while at the university level. So really just an academic background. Lived for a few years in Germany running a research group there before coming back here to the US and joining with Nielsen Labs. But my one true claim to fame is that I helped create a Mountain Dew flavor. This is Mountain Dew Whiteout. I don't know if you know about folks from Utah, but we love Mountain Dew a lot. There's a lot of shelf space in the store devoted to this. And because I helped to create the flavor, they put me in a Mountain Dew commercial. So that's the one cool thing that I've ever done in my whole life. So that's me. I'm part of the Toxicology and Chemistry consulting group at Nielsen Labs. So one of the reasons why I put this picture up here is it really kind of highlights how different disciplines that affect the eventual outcome of your medical device really need to interplay with each other. So the two people on my left, that's Dr. Campbell and Trevor Fish. Those are our toxicologists. So we sit in one room, we talk to each other all the time. On my right there is Thor Rollins. I don't know, some of you might know him. He's kind of like a godfather of biocompatibility in some ways. And it's not just because of his age or his receding hairline. It's more about, he's just so involved with these standards and things like that. And then, of course, there's me in the middle looking confused as everybody tries to explain things to me. But the point that I want to make here is that when we think about biocomp and toxicology and chemistry, sometimes these are a little bit too split up or discrete, when really, if you're going to do all of them or any of them very well, they need to have communication between each other. So if you're going to be doing chemistry, it should be informed by toxicology from the beginning, because what can happen, and what we see happen all the time, is somebody might do chemistry and take the chemistry results to us for, say, toxicology and biocompatibility, and the toxicologist will say, well, I'm sorry, these results aren't sensitive enough for us. I can't say anything. And if that happens, it's really inconvenient, right? Because you have to go back, test again, or find a low quality toxicologist. I don't know. So it's just it's better to understand or have a conversation with the toxicologist on the upfront and make sure that the chemistry targets that. It's also good to have biocomp folks as part of this conversation, because in the end, the toxicologist says, what can we do for chemistry? And this fits into like a bigger biocompatibility plan where overall we're trying to meet these regulatory requirements of the FDA or whatever the other reporting body is. So that's us. Just to give you just a brief outline, we'll talk a little bit about extractable leachables chemistry testing strategies and how these results are evaluated for regulatory approval. We'll talk about the way that we have been approaching this at Nielsen Labs. And again, this is an approach that evolves a little bit as we see the regulatory bodies, evolving as well. And then I'll focus on some of the changes that we see in the pipeline right now. Before I go even like one step further, I have to talk about E&L. I have to define it a little bit. So this is a little bit of a simplified diagram here, or maybe an idealized diagram. But when we talk about E&L, we're really, the E stands for extractables. And when we say extractables, we mean what could come off of the device or migrate from the device under exaggerated or worst case conditions. So exaggerated or worst case means at like higher temperatures than actual clinical use and using solvents and conditions that are more aggressive than clinical use. So we would expect that we would find, a wider variety of compounds and more compounds when we do extractables than when we do leachables. Leachables are what we get when we try as good as we can to mimic clinical use. So if we're talking about like an IV bag, this could be, that delivers saline, then the extraction medium would be saline, and if it sits there at room temperature, then the temperature would be room temperature, that type of thing. So, there are cases where, you might imagine that these two circles might not totally overlap, but if you design an extractable study, ideally, then they would overlap completely. So, I'm sure everybody here is familiar with ISO 10993 and this overall process, but just to put this into context here, how extractable leachables or E&L fits into this. So, of course, before a medical device can go to market, it has to be proven to be biocompatible, right? Per ISO 10993. And if you're a manufacturer and you have some new device and you want to bring it to market this way, you can demonstrate biocompatibility using a variety of different methods, right? So there isn't like a single prescribed, process there for you to get it to market. And some, processes or strategies are a little bit more burdensome than other ones, right? So, one thing that you can still do is you can do like the old school way, like from the 70s, and just straight up check box all of the risks with biological tests, right? So in ISO 10993-1 and in the FDA's guidance on the application of ISO 10993, it lists a bunch of biological risks. There's a test, like an animal test or a biological test for all of those risks. You can just run them all, and there's a good chance that this would get through. Another way to do it is to try to avoid testing to the maximum amount that you can, and take a look at the materials that your device is made out of and what you know about those materials, and try to explain why they don't produce those risks. And then finally, you can do E&L chemistry testing along with a toxicological risk assessment. And actually, in practice, the smartest way is, some combination of all three of those approaches. So that really frames things in a way, because the type of testing that we want to do, so, if we're going to decide what type of chemistry testing to do, we have options there too. There's like a whole field of different types of chemistry that you can do. And, this chemistry can range from quick and easy and not very expensive up to, very long and very, very expensive. And we have to remember that there's always a choice, right? If we want to do chemistry for toxicology and it becomes more expensive, say than running the animal test, then we shouldn't even try, right? Because it would make more sense just to run the animal test at that point. So to try to like frame that, I like to think about these E&L strategies on a spectrum where the x-axis here would be, say, predictive or scientific quality, and like the vertical axis is how burdensome it would be to the person that's sponsoring the study. So clearly, you would expect that for a higher scientific quality and predictability that it might get more expensive. And so if I try to like plot different strategies on this graph, I would put animal studies kind of like there. So to run the suite of biocomp tests that you need for, say, a permanent implant, you're looking at, I don't know, ballpark $200,000 worth of biological testing. But the scientific quality is good, right? And it's been good enough in the past, and it's still technically acceptable. But there's pretty strong evidence that shows that these animal tests aren't as good as doing chemistry in terms of sensitivity and predictability, right? So that's why I put it a little bit farther there on the left. Then when you start to talk about E&L, I imagine it really like this, where there's a lot of things that people call E&L, and it ranges over a pretty broad range of scientific quality and also cost. And you can do anything along that spectrum. So in this space, I would say that it only makes sense to like really think about E&L, and what we see from our side in terms of demand is when the quality of the science is better than the animal tests, and it's cheaper than the animal tests and faster than the animal tests, and the demand is pretty high, right? But as soon as the strategy becomes longer or more burdensome or more expensive than doing animal tests, then really the demand just drops to zero. Nobody wants to do it. And so this is where we see ourselves. So in the medical device world, we're trying to provide the highest level of scientific quality and diligence, while still being less expensive than animal tests. And certainly we would think about what needs to be done for pharma can be a little bit more burdensome even than for medical device, and that's up there. And this, the pharma techniques are still cheaper than, say, some human clinical trials and things like that. So along the spectrum, we have, say, at the low end of predictability or scientific quality, things like gravimetric NVR. As you move up from there, you go to these like non-specific, not super sensitive techniques like FTIR and TOC and so on. And then from there, you can add more and more specialized instrumentation or forms of analysis that increase the predictability and quality of the scientific data. And so really, if you go out and, say, shop for E&L work for a medical device, you could find anything, on this spectrum. And it's kind of up to, the person that, is ordering it, to decide what really you care to have included and what would your toxicologist accept and so on. So just to summarize, the goal of doing extractable leachables chemistry is to support device safety, right? So that's the end goal. We want to ensure that our devices do no harm. And really this is targeted in a sense where, in the FDA guidance and ISO 10993, for example, they call out specifically, like genotoxicity. So we do chemistry to say, okay, this device is not genotoxic, and we are addressing that risk that's identified by ISO 10993. So that's the goal of doing the testing. And, in the process, we're, saving time and money and reducing the burden of animal testing and so on. So, nobody really wants to just do like this repeat testing on animals over and over, right? Like the, titanium implants have been tested, thousands of times, the same tests over and over, and it just doesn't make any sense to like test it in an animal again when we have chemistry, that we can fall back on to make that prediction. Okay, so to sort of like frame this discussion about what are we actually going to do, I think it's good to start with like an example of a real device. So this is a medical device. This is a 3D printed Spock hand. It's intended to be implanted into the body next to the heart, so you can keep the memory of Leonard Nimoy with you always, right? So if you were the recipient of this device, right? So if you're the Trekkie that really, really wants this implanted, what sort of things would you be concerned about coming from the device from a chemistry perspective? Would you say, well, let's just do a gravimetric on this. Well, if it has 50 milligrams or less, then we're cool. Would you be like, well, it's plastic, maybe, let's just do like a specific plastics screen, like a plasticizer screen? No, right? So if it were me, and I do love Star Trek, I would be worried about anything that could come from the device, right? So what it has to, even things that we might not know beyond there, because even if I was like trying to target the chemistry testing towards what we would expect, I would wonder like, okay, well, what if there's like an oily leak from the machine that gets onto the device and some contaminant that we don't even expect would be on there? So for that reason, the only approach that makes sense is something that's very, very broad. So if you're going to do E&L to support biocomp and to do toxicology and for safety, it absolutely has to have a screening process. So there's no targeted list or database that will really do to really capture the risk there. So it's absolutely essential. It has a big, wide screening process. And so, and we think about it that way. We say, okay, we can't predict what might come off of the device. There's no way to know, like, what might have like dripped onto the thing, or like what was on the worker's gloves that touched the thing, or whatever. So we look for everything, and that's really the way that it should be. So that's frames the whole method that we're looking at. It's the same with. We keep that in mind when we think about extraction and all of like the sample prep for the devices. So for example, we have to pick solvents that will that we're sure can like solubilize many things. So we usually do a polar and a mid-polar and a non-polar, just to make sure we're covering the bases there. When we this is something that has come up more recently that I'm showing here. We do or can do this exhaustive extraction procedure. So this is something that, if I look at previous to 3 months ago and before, we never really did this, right? So it is, part of the standards, but it was pretty clear that it didn't make a whole lot of sense to do these exhaustive extractions from a toxicology perspective. We're seeing the FDA just demand this now a lot more frequently. So we'll do this exhaustive extraction procedure to try to make sure that we can, really recover all of the compounds possible from the device. This just involves serially extracting the devices over and over. And then if we think about the world of everything that could be on a medical device, and we say that we picked our solvents so that whatever we might think of could be found in there, then we need to start to think about analytical methods that we could use that are capable of detecting all of those. So chemists love to like categorize things. So I made this little like tree here. So if we think about everything out there, we can like roughly divide it up into inorganic materials and organic stuff. And then on the organic side, then we start to think about analytical methods that can, both do like this really wide screen and have the capability of identifying the compounds that are in there in addition to quantifying them. So that, meeting those requirements, the ability to screen, the ability to identify, and the ability to quantify really limits the types of analytical techniques that we can use to chromatography with mass spectrometry methods. So on the right-hand side, you see we have, roughly, very small molecules, and then like medium-sized molecules and large ones. And so by that I mean things that evaporate very easily, things that evaporate a little bit less easily. So something that would evaporate easy is like an alcohol or methanol, something like that. A medium-sized molecule might be something, more like a lightweight plastic additive, something like that. And then on the big side, these non-volatile organics, this could be some larger oligomers or plastic additives. things in that category. So for each of those, each of those categories, we really need a different method. So for the very small molecules, we use gas chromatography with mass spectrometry, but we do it in headspace. So it means that we either look for these volatile molecules above like the surface of the liquid that they're in, or in some cases I've seen people put like a whole device inside a jar and like measure directly over the device. For medium-sized molecules, this is where we really cast the widest net. So this covers easily 95% of what we might ever report for that. It's a direct injection into a GC mass spec. And then for the very big molecules, this is something that, it's listed in the standards, and that, depending on the situation, we may or may not include in the analysis. And this, is covering things on the very, really heavy end. So if there's not a risk of very large molecules coming out of the device, we might, not do LCMS. If it's like a soft plastic, then we definitely, would think about it. On the inorganic side, we can address that pretty well using ICP-MS. So that really covers a very broad spectrum. If you take a look at just the GC methods, using publicly available or purchasable databases, you can screen for roughly 400,000 compounds there. The lab that you use should have hopefully validated a pretty large set of common compounds out of that subset, so that you can get really good quantitative results in there. On the LCMS side, it's a little bit trickier because you can't have like a database, like a purchasable or publicly available database for LCMS. Is really instrument and technique specific. So the lab that you use should have built up their own database there to address those. Okay, so once you have your device, say it's the Spock hand, and you apply this strategy to them, you use a polar, a mid-polar, and a non-polar solvent, and you analyze by all of these different methods, then the end result is some sort of like chemical results like this, right? So, and this is really like the endpoint for E&L is to, with the right sensitivity, produce results, report it out in micrograms per device. This is what came from the medical device. And for, some folks, this might be enough, right, to stop there with the chemistry results. If you have a toxicologist on staff, or if you're, an engineer and you're really just kind of like looking for information about your materials, and that might be a stopping point. But more often than not, if you're doing this for biocompatibility, then you really need to follow this chemistry work with the toxicological risk assessment. And that's something that, a qualified expert should really do. So it's not just anybody can take a look at the chemistry results and say, oh, yes, this means it's safe, and have the FDA, actually believe them. So they're not like super specific about what a qualified expert means, but you really should be ready to defend that if they ever ask, right? So the toxicologists on staff at Nielsen Labs, so the Dr. Campbell is a board certified toxicologist. Trevor Fish has a master's in toxicology and is studying for the boards now. And this toxicological risk assessment, really what it does is it goes compound by compound and asks the question, okay, if this compound is really exposed to the patient and making a lot of worst case assumptions, would that be genotoxic? Would it cause chronic toxicity? Would it cause subacute, subchronic toxicity, and so on? Okay, so what I want to say here is that, if you're approaching this, if this is your game now, you have a device, or you're in charge of biocompatibility at your company or whatever, and you're going to like approach this, you have a device you want to get to market, there's a few things that you should really, think about when it comes to strategy. So first of all, the strategies are highly variable, as I said, and it can range from doing, basically no testing to doing everything. where the no testing, that involves maybe a couple of written documents that explain why you don't need to do any testing. Those documents can maybe cost a few $1,000s and take a couple of weeks to write, up to doing everything, where if you really do everything, you're looking at in excess of $200,000 in several months, right? So it's a big decision, and it's one that I think that you should start by having a conversation with your toxicologist, because you need somebody qualified to take a look at it and determine what really needs to be done. And if you're going to do chemistry testing, then that's especially important, because at the end of the day, the toxicologist is really like personally responsible for the results that they sign their name to, and they're not going to sign their name to some chemistry results that they don't have like full faith in, right? So start with the toxicologist. Once you know what the criteria are for whatever your toxicologist is going to do, then you start to ask yourself questions like, okay, to meet that, what solvents should I use, which ones, how many, do you really need to do this exhaustive extraction or not, like what's the regulatory risk if you do it or don't do it, and then you start to like think about the analytical methods that would be involved there to address all of those. So let's just, maybe it's easiest just to go through a couple of examples. So, here's a project that we worked on. And it comes up more or less regularly. So the device was an enteral feeding tube. It's designed to provide nourishment to underweight babies, so the population is neonatal. In this particular case, there was like several different tube material types, along with like a syringe for injection of the stuff, of the food. So the conversation started with the toxicologist, Dr. Campbell. Got her PhD working with in pharmacology, working with babies, so she's very sensitive and conservative, right? So from her perspective, the chemical analysis needed to be even more sensitive than what would normally be required, because she needed to make claims on developing, underweight, babies. And then, so that was her input. She was like, okay, we have to be extra conservative in this case. And then when it came to solvents, we looked at, okay, what do these materials really contact? And, we need to make sure that we cover those in the analysis. So the tube, goes through the infant's, nose, and then down into the stomach. And then there's obviously the food that goes in. The food is pretty fatty. So there's like, really the non-polar side of this equation is pretty well represented, as well as the polar side. So there's both, water and fatty stuff in there. So in this case, we did 3 solvents: water and IPA and hexane. We didn't see it necessary to do exhaustive extraction in this case, because the way that these work is that they're put in and the baby is fed over a period of time, and then the tube is changed. So we looked at that changing interval. And then, do you need fully quantitative results? So in this case, because the risk from our perspective was so high, we said, okay, yes, we really need fully quantitative results. So if we're going to base the safety of of an infant on these results, we need to know how accurate those numbers are. And that's exactly how we executed the study. So this is also an interesting case when we talk about testing strategies. So the company that made this device was taking a very conservative approach. They before they did chemistry, they did all of the required animal testing per ISO 10993 and submitted to the FDA, and the FDA kicked it back and said, yes, we see you did all the testing, and yes, you passed, but these animal tests really don't address underweight developing populations, right? These are like adult animals that the test is run on. So we don't really agree that this applies perfectly, and so they did the chemistry testing in response. And in this process, we actually identified, there were many compounds that came off of the devices. Always happens from plastics, but there was one that legitimately would have been dangerous to neonatal populations. So we caught a problem. The FDA was right. We were really glad that we were able to catch this, because we protected a vulnerable population there. And because we were doing this chemistry, we found this problem. It was identified. We knew exactly how much there was and what it was. The company was able to go back to the manufacturer of these parts. It actually came from the black rubber part of the syringe. They were able to go back to the manufacturer and reduce the amount of that compound that was used. They tested those parts again, and it was gone. So they were able to go to market with this device. So it's a great example of how chemistry can have a positive influence there and really be very useful when it comes to tracking things down. I mean, another example that we see a lot are things like wound dressings. So wound dressings are one of these things that you wouldn't think about really doing chemistry on, maybe just off of like the contact type and duration. But we've been seeing this more and more, especially where like the cumulative exposure of the dressing can go on for some time. But usually when we do these are to like a slightly different set of standards than the previous example, right? So here, if the exposure duration would be prolonged, that means that we're not like really worried about chronic exposures, and that means that the analytical evaluation threshold, or how sensitive the analysis needs to be, is quite a lot higher. When that's the case, things automatically become actually quite a lot less expensive and faster. So if you don't need to go down so low, there's a lot less time that it takes to identify compounds, and you don't need to go through extra concentration steps and things like that. So it really can save a lot of time to start this conversation in the beginning, think about how it's going to be assessed. So in just these last couple of minutes, I want to give you guys a chance to ask questions if you have any questions. Things that are happening now, as I said, this is a rapidly changing landscape. I find we set up a lot of these E&L studies at Nielsen Labs, and if there's ever like a question or concern from the FDA, then it's actually me that's on the phone with the FDA talking through it. So I hear a lot of feedback from them. So just to summarize what's going on now, so 10993-18, this is the section of the biocomp suite that focuses on chemistry testing. This has undergone like a complete revision. It's been in draft cycle after draft cycle, and it still is being revised and is in draft cycle. So hopefully, soon this is finished and everybody can actually agree with each other and we get this out. But you should be aware that this is coming. The new -18 really focuses on taking a risk-based approach to not only to biocompatibility but also to chemistry testing, which I think is a great idea, because we should. I don't really think that we should be doing a lot of scientific tests unless it makes good scientific sense in the first place, right? And so I think there's a motivation to do that. We've been seeing, speaking about doing things that don't necessarily make a lot of scientific sense. We've been seeing, a lot of demand for exhaustive extractions from the FDA, and so even though that's something that I don't necessarily agree with from a scientific perspective, we want to avoid the regulatory risk of asking them to, of having them kick that back. So, and this has been coming up over the past few months where they've really been wanting to see that for any permanently contacting device. So, so we're seeing that. We're also seeing a move to basically always include 3 solvents: a polar, a mid-polar, and a non-polar. Whereas before, if it was a type of device where it only contacted, say, saline, we would just use a polar solvent. Or if it was something like say a titanium knee or whatever, then we would do just a polar and hexane, a non-polar. But now they want to see a mid-polar. And then we're seeing an increased desire by regulators to see LCMS be included. In all cases, not just cases where there's plastics that it might present a specific risk. So with that, that's my last slide here. So my information is there. I love, like I said, I grew up under a rock. I didn't talk to anybody. It tickles me pink to just get on the phone and talk to somebody. So if you ever have any questions, please feel free to reach out and chat. I'm always happy to talk chemistry. I put some cards there in the back And I'm happy to answer any questions that you might have now. I think we have like 7 minutes if you have any questions that you want to talk about. Yeah.
So one of the big drivers for a lot of this regulation, whether it's UL 10993, is field failures, right? We know about chirality because we know about all these things because we've seen them in the happen, and we've seen things go awry. Especially with the new emerging field of biomaterials, where materials that we've seen are being processed in new ways, or they're new materials as a whole. Do you see a lot of internal and external pressure to go above and beyond what is the existing regulation, and if so, how is that handled?
So short answer, yeah. So if I understand your question correctly, is that we're doing some of this testing because there has been failures in the field, and sometimes people want to go above and beyond just to be extra safe, right? And especially with new things, and I think with very new things, that's a case where being more extensive certainly is justified. So. Short answer, yes. So we do see that. We do see people wanting to go above and beyond, and certainly, you take that on a case-by-case basis, and it's really the sponsor's choice, how far above and beyond they go. But I will say, in general, we try to discourage going above and beyond, right? Because if it doesn't make scientific sense or doesn't really address an actual documentable risk, then we worry that maybe we're leading the FDA in a way where they're going to be, less risk-based and just start to ask for more and more things routinely, even when it might not make sense. So you really have to balance that. When there's a real risk, though, especially with new materials, then we'll go as far as the sponsor wants, as long as it meets the requirement for regulatory concerns. I think it's a great question. Yeah. Any other questions or thoughts? No? I think we're all good. Okay. Well, my information's there. If you do have any other questions in the future, please don't hesitate to reach out. It'll make my day to talk to a living person. So with that, thank you.